Hi guys, hope you're all doing well. So I was doing a lot of research and I've been looking into the whole vaccine thing and everything that's been going on. Some of the stuff that I'm finding, I'm just going to read it. I'm not going to link it to anything. I'm just going to read this. I'm just going to call this something silly, this video. So this is a paper that I found called Monolithic Capillary Electrophoresis Device with Integrated Fluorescent Detector. So some of these papers are quite old. This one is from 2001 and it's talking about this, okay, which I've been talking about before. So I think the vaccine, obviously we know, has got a device in it, and there is something in the vaccine, and that's that. There's no playing around with that because there's too much information, too much evidence online. There's too much work that you can find to link this all together. I'll read a few bits to you. I don't know if this video will start. Hopefully it does. I'm not going to mention any of the main things. So a monolithic capillary electrophoresis system with integrated on-chip fluorescence detector has been microfabricated on a silicon substrate. Photodiodes in the silicon substrate measure fluorescence emitted from eluting molecules. The device incorporates an on-chip film, thin film interference filter that prevents excitation light from inhibiting the fluorescence detection. A transparent AZO conducting ground plane is also used to prevent the high electric fields used for the separation from interfering with the photodiode response. Separations of DNA restriction fragments have been performed in these devices with femtogram detection limits using SYBR green eye intercalating dye. Now, some of the things I'm mentioning in this piece, you go away and find out what they are and what they're used for, like I have. Now, this paper here is for the authors and who it's addressed to and what it is. It's from the Centre for Integrated Microsystems, Department of Electrical Engineering and Computer Science, Department of Chemical Engineering, and the Department of Human Genetics. Okay. Other things that are cited on here, articles cited and publications, some of them are improving the compatibility of conduct conduity detection with microchip electrophoresis using a bubble cell, label-free fluorescent assay coupled with reaction and SYBR green for detection of something. This is all to do with DNA research, guys. Some of the words I don't know what they mean and I can't read them properly, but I can study and find them out. Generation and manipulation of isotropic droplets in pneumatic medium using switchable dielectrophoresis. Study of peak broadening due to detection in the electrophoretic separation of DNA by CE and microchip CE, and the application of image sensor for ultra-small detection cell length. A fluorescent biosensor for detection of vital body fluids. Laser-induced fluorescent detection platform for point-of-care testing. Nutrient sensing using chip-scale electrophoresis. There's just so much here. Effective length collaboration method for processing the fluorescent signal detected by charge-coupled device in capillary. So, light-emitting diode, photodiode-based fluorescence detection system for DNA analysis and microchip electrophoresis. It's all here. Optimization of digital microfluidic biochip. What else we got? There's just a lot here. Obviously, I'll leave the link. Also, I found a I like to see who's been given contracts and stuff. This is a really good way of finding out stuff that they've done or that's been done or whatever. There's some old stuff from 2012 here I found, and 2013. And they're talking about microarray processing, and it's about electronic systems for DNA and RNA analysis. So the microarray processing, the process work is for genomic analysis in support of pulsed radio frequency RF bioeffects project. Specifically, the requirement for genomic analysis with support of subsection of the project, which is designed to investigate the potential for changes in markers in human T lymphatics that have been exposed to a pulsed RF signal. Data from this project will then be published to help influence the setting of exposure standards. Yeah, right. Detailed requirements is as follows. Analysis of the extracted mRNA 54 samples on an alignment 4 times 44K human genome microarray. This is required to provide continuity with previous data sets, provision of microarray reagents and gasket backing slides, quality control on received RNA using alignment bioanalyzer and nanodrop, microarrays to be scanned and on alignment G2565CA scanner, capable of 2UM resolution or better, data to be extracted using standard protocols and reagents, data experimental and quality control to be extracted, and there's more there. This is the next one. So this is the RNA and the DNA. The HPA unit based in requires an electrophoresis analysis platform, microchip technology, which is an alternative to agarose gels. The equipment must be able to analyze DNA single or multiplex PCR products, restriction digests, synthetic something such as primers and plasmids, RNA expression analysis, transcriptome analysis, quality check of in vitro synthesized mRNA in cell-free protein synthesis system, and purity checks on extracted RNA. The instrument must be equipped with an LED excited fluorescent detector 10 times more sensitive with ethidium bromide staining and therefore considerable safer, with no inherent GOSHH implications. After sample and microchip presentation, all preparation operations must be automated, including separation buffer filling, sample applying, electrophoresis detection, and microchip washing and data analysis. So half of that I don't understand, but I do understand most of it. And they're talking about what's going on today and what's happening. So I think that the people that have already had this are now readable. And the extra activity that we've been seeing in the sky, it's them reading the new systems. It is them reading the areas where people have been vaccinated, and they have been going around doing that and tracking it and checking the systems. That is what is happening. As crazy and as science fiction film this may sound, it is the real. I'm sorry, that's what's happening. So it's probably why so many doctors and nurses are aware, and they are saying no to this. You know, these are the people within this thing, and they're supposed to have witnessed so much of this pandemic, yet they're still saying no to something which the government is saying will protect them from this thing, but they don't want it, which is very telling. And it's very, very important that we start to spread this information and tell people. You mustn't be afraid to tell people. Just speak to strangers. It really, really does make a difference, okay? So guys, we don't want these systems in us. We don't want to be readable. We don't want to be fluorescent. We'll keep it 100% human. because you don't know when they can flip that switch and change those devices within those people. Peace out, guys.